Okay, so welcome back. This is part 2 of chapter 6, and we've just finished talking about transcription, and now we need to talk about translation. So translation is where we're going to actually make the protein. And again, just like when you've left your grandma's house and you're going home, you've got to get all the ingredients together to make the chocolate chip cookies. We've got to learn some ingredients here. And so the first ingredient that we're going to learn about is this one right here called the transfer RNA or tRNA. And there's a lot going on with this molecule, so let's take a look at this. The first thing that we have is we have this amino acid right here. Proteins are made up of amino acids, so the whole point of this is to gather all these amino acids in the right sequence. At the other end of this is something that your book calls the attachment site. I kind of don't like that term. I prefer the term anticodon. And the reason is because we see attachment site used throughout the book, like lots of things attached to lots of things. So anticodon is, for me, a preferable term. And I will probably be using that. I keep referring to attachment site, but I prefer the term anticodon. So anticodons are the complementary bases that match up with something called the codon in the messenger RNA. All that a codon is a 3-base sequence in the messenger RNA. Remember, the messenger RNA was the copy of the DNA, and it's that information that is going to be used to make the protein. But remember, it's the transfer RNA that's got the amino acid, and the amino acid is the building blocks of the protein. So how does this transfer RNA know that it's the next one in line? Because its attachment site or anticodon matches up with the codon in the messenger RNA. So there's actually this table of codons that we're going to become really familiar with. We'll use it in lab. We're going to use it in lecture / activities. So I just want to orient you a little bit to this, and then we're going to go back here and use this codon ACG to figure out how we get this amino acid just by reading the table of codons. So it was ACG, right? A was the first letter, C was the second letter. So there's ACG right there, and you can see that the amino acid name is threonine, which if I go back a slide, there's threonine right there. Sometimes we use the 3 letters, sometimes we use the whole word of threonine. It just depends on what you're being asked to do. So there's a couple of interesting things here, like here's one, AUG. AUG codes for the amino acid called methionine, but it turns out that methionine is always the very first amino acid that's put in every single protein. So we also call this AUG. We call AUG the start codon because it's always the first one. It is coding for an amino acid. And then we have all these different combinations here of 3 letters that we can get. But there's a couple of other really interesting ones, and these are called stops. Stop codons are what happens when the whole process of translation is stopped. And there's actually 3 possible ones. It could be UAA or UAG or UGA. Notice that there is no amino acid associated with them. So you don't actually get a last amino acid put there. It just says, nope, we're done, stop. So with start, you get an amino acid, but with stop, you don't. And then these are all the various amino acids that you can have. So if I gave you the codon of CAA, the first letter was C, and the second letter was A, so CAA is right here, and you would put glutamine in next. Glutamine, we also have something called glutamate, right? And those are actually different. So there's glutamate right there, and there's glutamine right there. Those are 2 different amino acids. So you will learn how to use this table during lab and lecture. So we've learned most of the ingredients from doing translation. One that we haven't yet met is the ribosome. We have already talked about the ribosome on several occasions, though. We learned about it in chapter 4, and that we've definitely talked about the ribosome. So we're going to learn a little bit more close details about this ribosome thing and how it all works together. There are 3 steps to translation. The first is recognize and initiate the protein process. And the way this works is that the mRNA comes in and it binds to the small ribosomal subunit. The small subunit and the large subunit together make up the ribosome. So 2 parts to this ribosome, the small subunit and the large subunit. So we get the messenger RNA binding to the small subunit, and then in comes this very important transfer RNA right here. It's actually called the initiator transfer RNA, initiator, tRNA. And it's always the tRNA carrying methionine that goes to the start codon. Do you remember what the start codon was? If not, you can rewind this video and go back and look that up. Or, I mean, I'll tell you, it's AUG. Remember AUG always carries methionine, transfer RNA, and it's the start codon. So here it's called the start sequence. So that binds. And then at the same kind of time, right about the same time, in comes the large subunit, plops on there, holds everything in place, and now we can start the next process. Do remember that whenever we have things matching up, anticodon or attachment site to codon, they're complementary. A's match with U's and C's match with G's. And there are no T's because there are no T's in RNA, and both of these are RNA. So that's the first step. We get everything coming together. The next step is called elongate. And this can be very confusing if you're just looking at a static picture. I very much recommend that you watch the animations in your book in this chapter. They are fantastic. I have links to those animations in Blackboard, or you can just go to the chapter 6 in Launchpad and watch those animations. So here's what happens. Our transfer RNAs, when they're done, they leave their amino acids behind. So you can see this one has left its amino acid behind. It's about ready to leave. In the meantime, we have our other amino acids they're coming in, they're reading these codons, and then they're attaching to, it's basically the last transfer RNA in the line, and then everything like the ribosome slides this way, or the mRNA slides this way, and we read the next open codon. So this is the next open codon. It's bringing in this amino acid. Everything's going to slide and engulf it and hold on to it, and then the whole sequence here of amino acid is going to pop over to whatever amino acid is in place next. And the same thing's going to happen as this one moves in here and the ribosome moves over. Everything's going to transfer up to there. So elongate is a confusing process. Please, please, please do yourself a favor and watch the animation on this. I literally can't lecture as well as that animation can help you visualize what's going on, because this is just a static picture. All right, so this is going to keep going until we get to the stop codon or stop sequence. And with the stop sequence, remember there's no amino acid, so it just everything just kind of separates. The large subunit and the small subunit pop away from each other. The messenger RNA is done because we've gotten to the stop codon. But what we have here is this newly formed protein made out of all of these little amino acids in a row, and now it's called a protein. The same mRNA, this mRNA right here, it can be read by hundreds of ribosomes. So the cell only has to make one RNA to make tons and tons and tons of those proteins. Hopefully this makes sense and you are starting to feel comfortable with it. If you're not, once more, please watch the video, the animation on Launchpad. So Remember that we talked about how every cell in your body, with the exception of those red blood cells, has the DNA for every single um, uh, protein in your body, right? That's gene expression. But what we need to talk about just really quickly is gene regulation, because we don't want eyeballs growing in our heart, nor do we want hearts growing on our head, right? They need to be regulated. Things need to be turned on and they need to be turned off depending on their location in the body, or whether or not your body needs to make this particular protein in this particular environment. For example, if you eat a lot of sugar, you need to make a lot of insulin. If you eat less sugar, you need to make less insulin. So that's what gene regulation is about. Control over whether or not the protein is made. And the environment definitely can have a lot to do with that. So one way that we can tell whether or not different genes are being turned on is something called a microarray, and we'll get a little bit of this in chapter 7 as well, but I thought I'd throw it in here to talk about how we can use the concepts of gene regulation to figure out what genes are being turned on or off. So we can put all these little pieces of RNA in these little plates here, and we can match them up with other pieces of kind of glow-in-the-dark RNA and figure out which bit of RNA we have. Is this the green bit of RNA or is it the red bit of RNA? I mean, clearly they don't have colors, but just visually helping you understand that each bit has been made by the cells, and we can literally, in a little plate, we can figure out which genes are being turned on in which areas of your body and which genes are not being turned on. Sometimes if a gene isn't turned on, it can lead to a disease. It could be a mutated gene. And so we have processes that allow us to kind of look at that. So what in your body helps control this? And the main thing is something called a transcription factor. And with transcription factors, what happens is that you get, if you remember the region of that RNA called the promoter. So I'm sorry, I said RNA, the region of the DNA called the promoter. I'm going to go back really quickly just so I can help you remember that. Remember that right there? That's where the promoter is in that DNA. When it comes to transcription factors, they actually help RNA polymerase bind to the promoter. Or alternatively, there might be like, nope, we don't want any RNA polymerase here, and they bind to the DNA and they stop that from being expressed. So we call that positive control or negative control. Positive control will speed up the initiation of RNA polymerase, and negative control will block or slow down the making of that polymerase, or the binding of that polymerase to the DNA. So that's one important thing. You get a lot of blocking of genes for eyeball manufacture in your heart, right? You get a lot of positive control of heart genes in your heart. So just a real quick look at it. I basically said everything, and I'm not going to go through these different regulators here. Just know that transcription regulates in positive or negative ways, and that you can actually also regulate after you've gone through transcription by quickly breaking down mRNA through enzymes, or processing that mRNA at different rates to make proteins. So positive control, negative control, it's kind of cool. It makes you who you are. So we are going to kind of wrap up the rest of this conversation talking about mutations. And mutations are any change to the base sequence of DNA. Look, That's the official definition, and that's the one that I want you to embrace. It's a change in the DNA. But having said that, you're just going to have to understand that you can use the term loosely to say that this is a mutant, but the mutation actually is in the DNA that's controlling this. And mutations can be bad, they can be detrimental. They can also be good. It's not quite as common to have good mutations, but they can be there. It's also fairly common to have neutral mutations. We don't think about them a lot. And we'll talk about the various ways that you can have neutral mutations when we get to lecture. Without mutations, and here's a key thing, without mutations, you do not get evolution. And we'll talk about that when we get into evolution a little bit more. So, so there are different kinds of mutations. There's mutations that happen in cells that are not your sex cells, like not eggs and sperm. If you have a mutation that happens in your lungs and it happens in your lifetime, you cannot pass that mutation on. If you get lung cancer because you smoked, your children cannot get that lung cancer because you smoked. On the other hand, smoking does also cause mutation in sex cells. So if your smoking causes a mutation in eggs or sperm, your offspring can get that mutation. Here's the really horrible part to think about. If that smoking causes mutation in eggs or sperm, you don't actually feel that mutation. Your offspring feel that mutation. And so that's kind of scary. That's kind of sucky, actually, that doing something in your lifetime, maybe you weren't even thinking about it, but that can detrimentally affect the next generations. And it's just the way it works, unfortunately. There's two basic categories that we're going to talk about for mutations. There's point mutations and chromosomal aberrations. Let's start out with point mutations. Again, we'll get a good bit of experience working with point mutations when we get into class. But if you think about it, there's three basic type of point mutations. And all that a point mutation is a slight change in the DNA sequence. So here's your DNA sequence. Remember that DNA makes messenger RNA, and that messenger RNA codes for proteins. So one thing that could happen is you could substitute out a nucleotide. So maybe instead of having a T right there, you're substituting in an A, and that causes a ripple effect. So nucleotide substitution can cause the amino acid sequence to be different, and you can see that difference right there. But it's only one amino acid. Everything else is exactly the same as it was up here, right? All the same. The ones that are kind of a little bit worse, even though they're just simple, they're just like one little thing, is a nucleotide insertion or deletion. So here we're adding in an extra nucleotide into the sequence, and that ends up because of our concept of the codon. Remember, the codon is 3 letters in a row of the mRNA. If you add a letter here, that actually shifts everything from there on. And we actually call this a frame shift mutation as well as an insertion or deletion, because the reading frame, the 3 letters in a row for the codon, shifts. So everything past the point of that mutation is no longer the same. This little substitution can cause problems, but this is huge. And so is a deletion. If you remove a letter from the sequence, everything kind of shifts backwards. You still have to read 3 in a row. And so every amino acid from that point on is not the proper amino acid. Again, a really, really big deal. Just think about it. If you've got a sentence in front of you and you add 1 letter and you're reading three letters in a row, everything else is after that point is going to kind of be gibberish. When it comes to chromosomal aberrations, we see kind of similar things here. We just talked about substitutions, insertions, and deletions. Well, with chromosomal aberrations, we're just talking about way bigger scale instead of just 1 letter. We're talking about like whole sections of DNA. So here is a gene that's been deleted. Gene number 2 has been deleted. If gene 2 has been deleted, we can't make protein number 2. We can't. It's no longer there. We can have something called gene relocations. So if you remove gene number 1 from this chromosome, but put it into a different chromosome, you can still make that protein. It's just in a different location. It can be a problem depending on where it gets put into the new chromosome. Finally, you can have something called gene duplications, and with gene duplication, we're seeing this with gene number 3 here. Instead of having 1 copy of gene number 3, we have 2 copies of gene number 3, and that, again, not a good thing, no bueno, because too much of a good thing is not a good thing, necessarily. Remember, please remember that, that most mutations are bad. Some mutations are neutral, some are good. We do have some gene duplications that are good, and we can talk about that in class. But again, usually too much of a good thing is a bad thing. What causes mutations? We've already talked a little bit about smoking, but smoking is called a chemical-induced mutation because cigarette smoke is a chemical. But not all mutations are caused by anything. In fact, a lot of mutations are called spontaneous mutations. These are happening just by accident during the process of cell division. And we'll learn more about cell division in, I think it's chapter 8. So those are called spontaneous mutations. We will bring up spontaneous mutations again when we get to chapter 8. But again, environment can cause problems. There's our chemical one right there with cigarette smoke. Radiation can also cause mutations. The radiation from the sun, if you spend too much time in the sun without any kind of protection at all, you get the sunburn, especially for pale-skinned people. And that burn there is just, it's not the burn itself that can cause the mutation, it's the radiation from the sun that causes the burn, but also causes the mutation, so sun's radiation. You can also be exposed to radiation through X-rays, and so this is a person getting X-rays from their dentist, and they've got this cover-up on their body, this lead cover-up, because radiation does not penetrate through lead very easily. So when you get X-rays, the expert, the person who's in charge of that will make sure that not only are you protected, but they're protected too. They walk far away from that. Maybe they duck behind a protective screen or so forth. That's the best thing to do because you definitely don't want radiation-induced mutations. So this kind of leads to a really natural question like, well, do sunscreens work to protect you from mutation? And we know that UVB radiation can actually damage skin cells. We actually know that UVA can also damage skin cells. Damage that DNA. So what sunscreens do are they're literally chemicals that absorb or reflect some of that UV radiation back away from your body, depending on the kind of sunscreen you have. These days, we think that the reflective kinds are the better kinds. So again, you might see people putting on like sunscreen that's really hard to rub in. Those are more of the reflective kinds. So lots of people assume that sunscreens are going to protect against skin cancer, but there's this really weird thing that's been happening because melanoma, which is literally skin cancer, has increased over the years, and so has sunscreen use. So there have been a bunch of studies published in the over the course of 20 years, probably back before you were born, that showed that there was this positive association between sunscreen use and cancer. So people started thinking that maybe sunscreen was causing that cancer or was not effective to protect you against UV. So everything, all those studies that kind of looked at that were all they're case controlled. They're looking at kind of case studies, and we did talk about case studies before. So they're just comparing people who have melanoma to people who didn't have melanoma and looked at whether or not they use sunscreen. So those weren't the best kinds of studies. Instead, what we started looking at is randomized control studies. So people were put into these different cases. This one group was asked to put on sunscreen. This other one was kind of whatever. If you want to use it, use it. But the first group was like, no, every day put on sunscreen. Just put on sunscreen every day. And there was and the other group was like discretionary. Oh, if you're going outside for a little while, maybe put on sunscreen, maybe you don't. It's totally up to you. So those were the two groups that they had, and they had 1,600 subjects, which is a really big case. In the regular use group, 11 people got melanomas over the course of that study. And this was a 10-year study, by the way. And in the discretionary use group, the ones that were like, whatever, use it or don't use it, it's up to you. Twice as many were developing skin cancers. And here's the thing. In these discretionary group, the invasive melanomas were 4 times more frequent. In other words, invasive melanomas, those are the ones that are the really, really bad melanomas. Those are the ones that will kill you. So what else did they look at? Well, they looked at time spent in the sun, and both of them spent the same amount of time in the sun. So what they've determined from this is that regular use of sunscreen, not occasional use, but regular use of sunscreen actually does help prevent skin cancer. It's just that you gotta be diligent about it. So, I mean, that's something good to take home. I want to wrap all of this up by talking about mutations and how this can affect diseases. When we spent a lot of time in class talking about cystic fibrosis, cystic fibrosis is not based on an enzyme. So let's think about how enzymes can lead to sickness. And remember, enzymes are molecules that make chemical reactions happen. So if you have a functioning enzyme, that functioning enzyme has the space in it, and that takes the molecule and makes the chemical reaction happen. If you have a mutation in your DNA, your enzyme doesn't have the right shape to it, so it's not going to allow that chemical reaction to happen. And so what happens is you get a buildup of this chemical here. And this buildup of this chemical can be really, really bad. It can be fatal. There's some, like Tay-Sachs disease, which is where your lysosomes, if you remember, we talked about this before, where your lysosomes aren't working. If your body's not getting rid of waste products, you're going to die. On the other hand, maybe you have the disease called fast flushing. And fast flushing is what happens if you drink alcohol and you don't have the working enzyme. If you drink a little bit of alcohol, your face flushes and you look really red in the face, and you might feel very sick, you might throw up, but you won't die. If you drink a lot of alcohol and your enzyme's not working, you can actually die from that. So this is just something to keep in mind, how important enzymes are and how important mutations are to the functioning or non-functioning of the body. Well, that's all I have for you for Chapter 6. That's all I have for you for Chapter 6. Hope it was useful, and I will see you in class.